Welcome to chapter 14. In this chapter, we're going to talk about inheritance. And we're going to take a step back about 140 years ago, back when Gregor Mendel, this Austrian monk, started doing some crosses with peas. He was a very educated man. He loved his two loves of science were math and plants. And so he started doing these crosses, and some of the observations he made with his crosses, it should be noted that were the same observations that had been made for many years. What Mendel did that was special was he started to apply numbers to them so that it started to make sense. And by applying these numbers, he was able to come up with his two famous laws, the law of segregation and the law of independent assortment. So let's take a step back and look at some of Mendel's crosses. In the book, it talks about purple flowers against white flowers. So let's talk about something else. Let's talk about green seeded plants and yellow seeded plants. And both of these were what Mendel would have called pure breeding. And what do I mean by pure breeding? Pure breeding means that when you take a plant with a particular trait, for instance in this case yellow or green, when you take a green seeded plant and you cross it with another green seeded plant, you get all green seeded plants. It's a pure breeding strain for this trait. The other traits in this plant might not be pure breeding, but for seed color it is. Same thing with yellow. You take a yellow times a yellow seeded plant, you cross them, and you only get yellow. So what Mendel did was he crossed these two plants, a yellow seeded plant by a green seeded plant. Now there could have been several predictions from this. You might predict that you'd get all green, so maybe all green seeded plants. You might also make a prediction that you'd get all yellow seeded plants. So this is our second prediction, all yellow. You might also predict that it would be a mixture, some green seeded plants, some yellow seeded plants. So let's symbolize that by just putting a plus here. So green seed plants and yellow seed plants. Finally, you might make a prediction that you would see you would see seeds that were a blended color between green and yellow. And so let's draw it like this, because I don't have a color in my choices to blend two colors. So let's just draw a seed that's kind of partly green and partly yellow. Now, when Mendel did this cross, he didn't see all green. He didn't see a mixture of green and yellow. He also didn't see a blend of green and yellow. He saw all yellow. In fact, it should be clear that Mendel was not the first person to ever do this. People knew for a long time if you took a yellow and a green seeded plant and you cross them together, you'd get yellow. What Mendel did, because of his love of numbers and math, he started applying math to these rules, to these observations. That's what made Mendel's observations special, which led to his two laws, the law of independent assortment and the law of segregation. Let's also back up and define some terms. This first generation here, the two that started it, we call this the P generation. That's parental. When the two parental generations have progeny, that progeny is called F1. F stands for familial, so the first family generation. Now what Mendel did was he took these two yellow plants and he crossed them to each other. Now remember, if you take two yellow pure breeding plants, seeded plants, you're only going to get yellow seeds. But these aren't necessarily pure breeding because they just came from this generation here. So let's take another yellow seeded plant and cross them together. So you take a yellow seeded plant times a yellow seeded plant, what do you get? Well, you might predict you'd get all yellow, you might predict you'd get green, you might predict you'd get some kind of blend. But what he saw was something different. He saw some of the, let's make this yellow. He saw some of the plants that were yellow, rather they had yellow seeds, and then we'll get rid of this black line here. He also saw some plants that had green seeds. Now, again, people had known this before. This was not the first time this was ever observed, but what was special about what Mendel observed was the numbers. Every time he did this cross, and he did hundreds of these, as a monk he had a lot of spare time on his hand, and he did a lot of these crosses. And every time he did these crosses, he saw approximately 75% of the plants had yellow seeds, and 25% had green seeds. I should also point out that this generation here from the F1 is called the F2. So let's put that down so we don't forget to do that. Every time we saw these numbers, and being a person who loved numbers, this particularly intrigued Mendel. Why are we always seeing these exact same numbers? This led to some of his predictions, or rather I should say some of his what we call concepts. The first is that these traits, yellow or green, or any of the traits he looked at, had to be determined by things called genes. He didn't call them genes. That term came much later. But they had to be caused by genes, and there had to be alternative forms for these. For instance, like a green color seeded gene and a yellow colored seeded gene. So let's go ahead and write that. Let's go and write that here. 1. And we now know that these alternative forms of genes are called alleles. So let's just put genes exist as alleles. That is to say, every gene, there has to be alternative forms of genes that give you these different kinds of phenotypes. The only way these numbers work out is if his second prediction here is true, his second concept, and that is that these alleles, and again, he didn't use the word allele, that's a term that came much later, but since that's the term we use now, let's go and use it in our discussion now. These alleles, in every organism, they must exist in pairs. So every organism, for any gene, they have to have 2 alleles. This makes a lot of sense now that we understand chromosomes. Mendel didn't understand chromosomes. They hadn't yet been discovered. But we know we get one set of chromosomes from mom, one set of chromosomes from dad. Let's just draw a chromosome here, because drawing chromosomes are fun. So this one comes from mom, and this one comes from dad. We know this chromosome, each of these chromosomes may have thousands of genes on them. So we have a gene here and a gene here. Each comes from mom and each comes from dad. We call these 2 genes we inherit from our parents as alleles. If we had more than 2 alleles, these numbers wouldn't work out as well as they work out. If you had 4 alleles, these numbers wouldn't work out. Now his other prediction here is often referred to as the concept of dominance, because it deals with dominance. So I'm just going to write concept of dominance here. And then I'm going to explain what I mean by that. The concept of dominance. What I mean by this is anytime you have 2 alleles, in this case the allele for seed color, you have a green allele and a yellow allele. One of them will determine the trait. That is, one of them will act dominantly over the other. The one that acts dominantly over the other is called the dominant allele. In our example here, yellow is dominant. So up above by yellow here, we're going to call yellow dominant. And the only way that you'll ever see the allele that's not dominant expressed as its phenotype is if it's the only one present in the organism. So we say that allele is recessive. And you only see a recessive allele presented as a phenotype is if it's the only one present, if both copies are recessive. The dominant allele exists, will give you the phenotype as long as one of the 2 copies is the dominant allele. So in this case, as long as one of the 2 copies is the big Y allele, then you're going to have a yellow seeded plant. Okay, now his fourth concept here, and this ultimately is his law of segregation. Let me write this down and then I'll explain it to you. And what the law of segregation means is that you have 2 alleles for any given trait, and they separate or segregate away from each other during meiosis. Or what Mendel would have said is that they separate from each other during gamete formation, but we now know that that is meiosis. So 2 alleles separate from each other during meiosis, and each of those alleles will end up in a different gamete. I'll say it again. The 2 alleles for any given trait during meiosis will separate away from each other, and when they separate away from each other during meiosis, they will end up in separate gametes. Now over here, let's draw the alleles next to these pictures we have here. And what we need to do is identify our allele. So let's identify our allele as big Y. That's our dominant one, and our recessive one is little y. This dominant pure breeding yellow seeded plant we're going to call big Y, big Y. Our recessive pure breeding green plant is little y, little y. This illustrates his first two ideas here. Genes exist as alleles. Our alleles are Y, big Y, and little y. And every organism has to have two of them. So the yellow seeded plant has two alleles of big Y, big Y. The green pure breeding seeded plant is little y, little y. So our first two ideas here are taken care of. When we cross these two plants, they are going to separate from each other during meiosis to make separate gametes. So this is his final law here of law of segregation. We'll come back to dominance in a moment. But we know when this plant here, the big Y, or the yellow seeded plant, goes through meiosis, it's going to make one gamete that's big Y, that has the big Y allele in it. It also has all the other alleles of all the other genes, but we're just focusing on the coat color, the seed color, right now. So it will make two gametes, each with the big Y allele in it. Again, it's because during meiosis, those chromosomes which contain the genes segregate away from each other into these two separate gametes. Same thing with the little y. So you're going to end up with a little y gamete in this one, and a little y allele in this gamete. Please don't mistake the Y for like the Y chromosome that determines maleness. We're just talking about a single allele, a single gene. All right, so now these come together, you cross them. We know that the progeny now are going to contain one gamete from this parent, so it's got to contain the big Y, the zygote that's formed, the embryo that forms, and the other parent has to contribute one of its gametes, little y. Now when you look at these gametes, when you look at these pea plants, you determine that these indeed are yellow seeded plants. They contain one big Y, one little y. This comes back to our idea of the concept of dominance. As long as one copy of the pair is the dominant allele, in this case the yellow big Y allele, then you will express that phenotype, the yellow seeded phenotype. So the concept of dominance again says as long as you have one one dominant allele, in this case it's big Y, then you will express the phenotype associated with that dominant allele, yellow in this case. All right, now how does the law of segregation explain this 25% to 75% numbers for the F2 generation? Well, remember, you're going to take this plant here, this big Y, little y plant, and what you're going to do is you're going to cross it to itself. This plant here, half of its gametes will be big Y, contain the big Y allele. Half of the gametes will contain the little y allele. Same thing with this plant here. When it produces gametes that are going to cross with this plant here, big Y, little y, in separate gametes. Separate gametes because when the chromosomes segregate, they each go to a different gamete. All right, now, we know that the progeny from this plant here, remember 50% are big Y, 50% are little y. So we know 50% of the progeny are going to get the big Y here. So let's just write big Y. 50% of the progeny are going to get this little y. So let's write little y here. We know when it crosses with this gamete that has big Y is just as likely to get this big Y here. So we'll put big Y here. And that would be one progeny. This little y could either get the big Y, so we'll go big Y here, or this little y, we'll draw it again here, could also get this little y. It's just as likely. Random fertilization, meaning that the gametes from this organism are just as likely to pair up with either one of these. Now, we've missed one here. This big Y, remember I said it could pair up with this big Y, but that big Y on the left there could just as easily unite with the gamete that has the little y. So each of these progeny are just as likely in the F2 generation right here. Big Y, that is formed with this gamete here, and this gamete here from the other parent. Little y, big Y, that's formed because of this gamete here, and this gamete here. You could also have little y, because it could just as likely, through the random fertilization, receive the gamete with the little y allele, or this, and then the other organism could contribute the other little y allele. Then finally, we know that an embryo could be formed from this gamete carrying the big Y when it fuses with the little y gamete. Each of these organisms, big Y, little y, big Y, little y, big Y, little y, are equally probable. You would expect a 1 to 1 to 1 to 1 ratio of each of these. We call this the genotypic ratio. The genotype are the two alleles, big Y, little y, big Y, little y. These are, or big Y, little y. These are the 4 possible genotypes. Genotypes only refers to a single allele pair. So if on a test I asked you to list the possible genotypes from a cross such as this, you would write big Y, little y, big Y, little y, big Y, little y. That is the genotype. Now, in fact, why don't we just write genotype, since let's see, can I write it down here somewhere? We'll write it here. Genotype. Now, how that genotype is manifested into a visual trait is dependent upon the concept of dominance. Okay. As long as you have one dominant allele, this one has a dominant allele, this one has a dominant allele, and this one does. All 3 of these, so we're going to have 3 yellow seeded plants because of dominance. And the only way you're going to see a green seeded plant is as if you have the 2 recessive alleles, in this case little y, little y. And we only have one of those options, so it's 1 green. So it's a 3 to 1 ratio, 3 yellow to every green, or 75% to 25%. We call the manifestation of this genotype here the phenotype. So let me write the word phenotype now. In this case, the phenotype is the color of the seed, yellow or green. Okay, the other 2 concepts I want to make sure that are clear are the terms homozygous and heterozygous. Okay, if we're going to use the same terms we had on the last page, or same letters, a homozygous individual would be something that has a genotype with 2 of the exact same alleles. So big Y would be homozygous. Homozygous dominant. Or you'd have little y, which would be homozygous recessive. Heterozygous, hetero meaning different. You'd have one of each allele, one dominant, like big Y, and one recessive, little y. So those are 2 very important terms. Now, on your study guide, I mentioned that you needed to know 5 steps in order to do any genetic problem. And I want to review that real quick with you. And in doing so, let's talk about a human condition like, say, cystic fibrosis. The first step in doing one of these genetic problems is to determine which form is dominant and which is recessive. Dominant, so I'm just going to DOM versus recessive. All right, well, we know that a condition like cystic fibrosis is a recessive condition. So let's identify our terms. The dominant allele, let's make that big C. And a recessive allele, let's make that little c. Now let's say, let's take a step back. So we know that anybody, let's take another step back and just review something. The dominant allele is the big C, recessive allele is little c. The way you see a dominant phenotype is as long as you have the big C there, it doesn't matter what the other allele is. It could be a big C, or big C, little c, and you will have the dominant phenotype, which is not having cystic fibrosis. The only way you see cystic fibrosis is if you're little c, little c. Now, on a test, well, I'm sorry, let's see here. The next step is to determine the genotypes of the parents. So let's just go geno of the parental generation. So genotype of P. All right. Now, in any given problem, there might be a little scenario presented to you. And maybe in this scenario, we're told that each of these parents had a healthy parent, somebody without cystic fibrosis, and a parent with cystic fibrosis. And if that's the case, and the parents we know, the P generation, the story tells you that they don't have the disease cystic fibrosis. So we know that each of these parents would have to be big C. If we knew one of their parents had cystic fibrosis, one of their parents was little c, then we know that they too also have one recessive allele. So to review some terms, each of these parents we now know is heterozygous for the cystic fibrosis allele. But they're healthy because of the concept of dominance. Big C, as long as that big C allele is there, you're healthy. All right. Then the next step is determine the gametes from each of these parents. Let's just make this one dad and this one mom, just for the sake of argument. We know that dad will produce gametes that are big C and gametes that are little c. Let's circle them here. And we know mom will do the same. Big C, little c. The next step is to put the gametes in a Punnett square. We haven't talked about Punnett squares yet, but a Punnett square is simply this. Draw our lines like this. Mom's gametes will go on top here, big C, little c. Dad's gametes will go along the vertical, big C, little c. We know that each of these gametes from dad equally probable of producing either one. Same thing with mom, equally probable of making either one of these. So we put them on these different lines, which we call the Punnett square. And now we unite the gametes. Big C from mom is just as likely to unite with big C from dad, or big C from mom with little c from dad. We know that the big C here from dad could also combine with little c from mom, or the little c from both of them could combine. Now, having this, it's easier to calculate our frequency. Our genotype frequency is going to be 1 big C C to 2 big C little c's to 1 little c. And then our phenotypic, so this is step 5 of this process. I should put that here just so you're following along here. The phenotypic frequency is 3 individuals, this one, and this one, that are healthy. So I'm just going to put big C, little c. They're healthy. 3 unaffected individuals to 1 individual, this individual here, who has cystic fibrosis. Genotypic frequency is 1 to 2 to 1. Phenotypic frequency is 3 to 1. All right. That ends the first part of Chapter 14. I'll put another podcast for the last part of Chapter 14. And that'll end the material for this week before the exam. Let me know if you have any questions, and until then, I'll see you in class.